All right, well, thank you for joining. We're going to talk today a little bit about some of the technology that is available in the area, specifically of urine drug testing. It's certainly something that can be taken outside of that space, but we're really going to focus on this particular area and see if we can't figure out where technology belongs in this area, where we've been, and where we might potentially be able to go with this idea of the necessary evolution of urine drug testing. I'm Dr. Frederick Strathmann, Medical Director of Toxicology at AACC Laboratories. And again, thank you for joining today. So first, the current state of urine toxicology testing is important to think about. So most of us who oversee any type of urine toxicology testing probably have any one or multiple of these currently in our laboratory. So we may have immunoassay screens only, and that may be because we don't perform confirmatory testing in our lab. It may be because we're trying to offer screen-only assays for individuals, physicians who are looking for that option for whatever reason, whatever application they might be thinking about. And then certainly many of us are also aware of immunoassay screen with reflex to confirmation, which is really sort of that tried and true workflow that a lot of us are familiar with. Where we have that immunoassay screen, a lot of times that's considered presumptive. And then we move on to confirmatory testing for positives, either by liquid chromatography or gas chromatography mass spectrometry. And then certainly one of the workflows that has been growing in popularity, arguably a GC mass spec assay for comprehensive testing has been around for quite some time. But this idea of going direct to mass spec, not doing any presumptive testing, not inside the forensic area, but certainly outside of that in the sort of classic urine or clinical toxicology area, is something that has been gaining a lot of favor and continue to do so as well. And then certainly there are other approaches that we have explored at AACC, such as the hybrid model, where we have high-performing immunoassays, so immunoassays with very low false positive rates, but at the same time adequate sensitivity for what we're trying to do. And we couple that with mass spectrometry for immunoassays that would normally not perform well. We move all of those drugs and metabolites to mass spec, but keep the immunoassays that perform well. And the goal behind all of these is really trying to adapt to what the environment is changing. So are we looking for rapid turnaround time? Are we looking for definitive testing? Are we trying to keep costs down? And certainly we're trying to do almost all of those at the same time. But again, the clinical context is what's really going to dictate what we're trying to do. And then certainly there's a lot of technologies that have made their way into the laboratories because of toxicology. So things like immunoassays, which many of us are familiar with, anywhere from automated platforms to ELISA-based testing, or even point-of-care devices as well. Quadrupole mass spectrometry, by far the most popular type of mass spectrometry that you'll typically find in the clinical laboratory. And even though the technology itself is a bit older, we're starting to see the entry of time-of-flight technology into the clinical laboratories as well. And then sort of a best of both worlds to some degree, a quadrupole time-of-flight technology also in regards to where that technology might fit and how we might use that. So I mentioned already a little bit about a tried and true workflow. And so this is really what most of us, I think, are used to, is we have a sample that's collected. We send this in for most often a screen with reflex is ordered, any one of the number of panels that a laboratory internal or external might have on their menu. And then it's really a decision of, is the result negative or positive? If it's negative, typically the testing algorithm stops there and you report out a negative. We're all sort of very familiar with the concept of false negatives, however, and certainly we cannot reflex every result, whether it be positive or negative, to confirmation. And so for that reason, a lot of times we do bear the brunt of false negatives and needing to follow up manually on those, having physicians to call in and add on testing if they're wanting to follow up on an unexpected negative. On the other side of this, we have a very common sort of positive result by immunoassay screen. We reflex that to mass spec confirmation with the idea that the mass spec confirmation is oftentimes more sensitive, but more importantly, it is more specific, and that's really what we're looking for from that side. And then based on the mass spec confirmation, if it's positive, then the immunoassay result itself was also positive and was not a false positive. And so for that reason, we report out typically a concentration, although certainly you don't have to, only because you're using mass spectrometry. And on the other side of that is if we generate a negative result by mass spec, then the assumption is it was a false positive by the immunoassay or the screening method. And so for that reason, we report out a negative, and essentially that negative confirmation result trumps that earlier immunoassay positive result. But that can still cause some problems depending upon how that screen with reflex workflow is being interpreted. So we certainly do have some challenges with immunoassays, and that's one of the reasons why we're talking about the different types of technologies that is available. I do want to stress that not all immunoassays are created equal, just like not every mass spec or mass spec assay is created equal as well. But certainly here in the example we're looking at, we have an immunoassay that is designed to detect the opiates class. And as most of us know, whenever you're looking at what an immunoassay is going to cross-react with, there's always sort of that surrogate or that sort of typical target, and then it's a degree of cross-reactivity that you might have for everything else. So in this situation, and in most opiate assays, we have morphine as the typical target, and then we have varying degrees of cross-reactivity with things like 6-monoacetylmorphine or a heroin metabolite, hydrocodone, and codeine as well. And now the real downside to immunoassays is you could argue that this immunoassay itself is actually quite specific for opiates. But again, the interpretation becomes problematic because it ultimately ends up in a signal, but it doesn't really differentiate necessarily what we're looking for. So the final interpretation of this sort of summation of a signal and a positive result from this immunoassay could be interpreted as morphine, which would be completely appropriate. It may also be 6-monoacetylmorphine or a heroin metabolite, which is obviously completely different than if we're prescribing somebody morphine or we're prescribing somebody hydrocodone. And what's really cross-reacting is morphine, but 6-monoacetylmorphine, indicating that there's some heroin use as well. So the problem really comes into is we can't really fundamentally answer, is this all of the drugs that are in there, or is none of them in there, and it's something that's completely cross-reacting that's not even in the opiates class. And so classically we get a result, but we don't always get the information that we need, and so this moves on to confirmation. So highlighting a little bit more of some of the problems with immunoassays, a recent paper by Dr. Johnson Davis, one of the medical directors at AACC, summarized results from immunoassay screens that we have internally that are very commonly used as well, looking essentially at what the positivity rate was, but more importantly, what I've highlighted here is the false positive rate as well. And as you look down through this column, you can see a wide range of false positive results from 100% false positive results for things like MDMA and PCP, and going all the way down to 0% for immunoassays that I would argue a lot of people are very confident with. Cocaine is one that is typically has an exceedingly low false positive rate, and I think the data here indicates that also. But again, even the immunoassays that had low false positives, so a good example again is a benzodiazepine immunoassay, we still don't know which benzodiazepine is actually there. We know the class, but we don't really know the specific benzodiazepine, and so to some degree we haven't really gotten all of the information that we need. But overall, what I'm highlighting here is we have immunoassays that have up to 34% false positives at times, depending upon what it is we're actually looking for, and that can be a huge problem overall. So the other problem that comes in with this also is the concept of panels. And this is something that I think every laboratory struggles with, every physician struggles with, and it's this concept of one size doesn't fit all. We certainly would want it to. Many of us have very classic sort of drug screen panel 9s or sort of our core drug screen. And here what I'm highlighting is 3 different laboratories with published inclusion lists for their immunoassay screens. And you can see they vary quite a bit, but these are each laboratory's sort of core immunoassay-based screen that would reflex to confirmation. And so this can be challenging, obviously, for a physician who's looking for the right test and the right fit, but it can also be very challenging for the laboratory trying to continuously develop different combinations of panels in order to try to accommodate everybody's interests and needs. And of course, that doesn't take into account that if we are doing an immunoassay-based screen, we still do have fundamental flaws with the majority of the immunoassays also. And so this is where we get to the point of having expanded screens. And so we have more compounds added to this, so that way we can try to catch a larger audience. So rather than having to have lots of little panels, we have one big sort of mega panel or big panels that we put together. But the problem is always that something is going to be missing, right? There's always going to be that request for pregabalin or gabapentin, and we didn't put that into our screen. So then we have to find that assay, bring that assay online, then put that into our panel. And then, of course, the continued sort of onslaught of requests because one size doesn't fit all, and that does become the problem. And so ultimately, something is always going to be missing, no matter how big the panel we ultimately put together. And then ultimately, we're looking at how big of a panel should we really be putting together to be cost effective, but also thinking about utilization and what we're trying to do. So the flaws with the immunoassays themselves in terms of the method, but then also this limitation in panels has led to an increase in the number of mass spec-based screens. And I still use the term screen here because really what we're looking at is this concept of we found the results initially, but we don't have a true confirmation because we're not reflexing them to another method to find them. Arguably, mass spec is definitive, and so you may say that we don't need to reflex them to a more specific method. But there can be power in having sort of one sample preparation and another sample preparation. So I just want to be clear here that we are talking about sort of breaking this kind of screen with reflex, going straight to the mass spec test, but we can't really term that as confirmation because we're not confirming anything. So we have samples collected, we do a mass spec based screen. Then a nice thing that we have here is we can still have things reported out as positive, negative. We can obviously report them out quantitatively as well. There's no magic to reporting qualitative versus quantitative as long as you're controlling the assay appropriately and have the appropriate set points in there. But one of the things that we have here is we have much better specificity when we're looking at these things. And so for that reason, why is mass spectrometry so specific? And we have different types of mass spectrometry that are available. Triple quadrupole by far is the most common type of mass spectrometry in the majority of clinical laboratories doing sort of classic confirmatory or definitive type testing. And here we have 3 different quadrupoles. Sometimes, obviously, the second quadrupole is not really a quadrupole, but maybe something like a hexapole. But essentially, the idea is we're looking for a precursor mass. We're going to fragment that, and then we're going to look for one of those fragments, and oftentimes multiple of those fragments in that final quadrupole. And what that allows us to have then is an enormous amount of specificity because we not only have to have the right precursor mass, but we also have to have the right fragment. And then we can take it a step further. And not only do we have to have one right fragment, but we can have 2 right fragments, and we need to have those fragments in the right ratio. So we're getting to orders of magnitude when it comes to specificity over top of the majority of immunoassays. You can argue sensitivity depending upon the immunoassay itself. Some are actually quite sensitive overall, especially when they happen to be quite specific, but cross-react very closely within a certain classification of drug. And then on the bottom, I'm kind of showing a very basic schematic of time-of-flight mass spec as well. Slightly different physics, of course, but ultimately what we're trying to do is the same thing, is we're using mass spectrometry in order to measure a molecule's mass, in this situation, a drug or a drug metabolite. And there are advantages and disadvantages to each one of these methods. And one of the advantages that I will bring up now that will come back a bit later is that with a quadrupole, we're always sort of tuned to a specific mass to charge ratio, or in most instances, a specific drug that we're looking for. So at any given time, we're only looking at morphine, and then we need to sort of switch the settings and the parameters of the mass spec in order to look for a different molecule. And so you can sort of immediately understand that the more complex the analysis you're trying to do, the faster the analysis, you do start running into the inability to measure enough of each one of those compounds you're looking for, and that can lead to specificity issues and poor data quality. On the opposite side of that is time of flight, where we really don't have, for the most part, any kind of data loss. That's not entirely true, but we can kind of think of it that way for illustration today. And instead, we line everything up at what is essentially a starting line, give everything the same sort of voltage push, and then we just wait for everything to eventually hit the detector. Depending upon how long it takes to hit the detector is correlated to its mass. So for that reason, we have a much wider or a much higher ion transmission with time of flight. We don't have to do the parameter switching and setting. So instead, we pulse ions through and we collect them all. So that does have some capabilities when we're looking at very fast methods, or areas where we have extremely complex methods and we want to be able to sort of measure a lot of molecules in that single amount of time. And we'll come back to that concept. So some of the benefits then of a mass spec-based screen, and there are a lot, and that's why a lot of organizations are moving towards the idea of a mass spec-based screen. So you have sensitivity and specificity that's on par with what have been referred to as classic confirmatory methods. And in a lot of ways, we're using the exact same technology that we always did for confirmatory methods. At the same time, if you have sort of that initial screen being done by mass spec, you can typically see individual compounds and metabolites. So not only do you get to see oxycodone, but you could add oxycodone as well as noroxycodone. You get a lot more information than you get from a positive oxycodone immunoassay, when you may not be able to tell the difference between addition to the urine and true ingestion of a drug. We can eliminate cross-reactivity complications that we have for immunoassays, so we don't have to catch people who are trying to quit smoking and thinking they might be abusing amphetamine. So drug metabolite pairs for interpretations can be there as well. Drug abuse testing can be done concurrently, so it's not as if we can only do oxycodone and its metabolites. We can do a lot of different things by mass spec, depending upon what we want to try to package together. And you can argue that if you've got the technology and the expertise to be running mass spectrometry, then integration of new targets is really not that difficult. You don't have to wait for, at least right now, we don't have to wait for FDA approval. We don't have to wait for a kit to hit the market. Assuming there is a standard and we can develop a method to high quality, we can put that onto our test menu as soon as we'd like. And then certainly if we decide to go down the qualitative route, and I won't get into too many details with that, but again, reporting quantitation versus qualitative results, there's a time and a place for both. And so we can still have a reflex to confirmation, although we certainly need to be careful based upon the technology we're using, what codes might be available for reimbursement in the future. So a lot to take into account when we're trying to think about designing the best workflow for proper utilization, but also for what physicians need to be able to interpret what they're looking for. And I just wanted to highlight, I'm not going to go into the details of quantitation versus qualitative results, but we did recently have a paper published in the Pain Medicine Journal looking at the cost and efficacy assessment of an alternative method that we used, reporting qualitative results. I think it's quite interesting. I think the punchline from it is about 3% of results required quantitative confirmation in order to appropriately interpret. So it kind of gives you an idea of how valuable or lack thereof there is for quantitation, but it certainly does have its place. So the problem we still run into, however, is even if we're using a mass spec, we have a very specific method, we've got great sensitivity, we don't have to worry about confirmatory and follow-up testing, we still have this problem of panel size that we need to hit. So one size still doesn't fit all. You could argue that doing a larger panel by mass spec is a little bit easier than potentially trying to do it by immunoassay. But at the same time, you still have the issue of high positive rates in certain compound classes and specific compounds. And then you have other compounds that are kind of just along for the ride. You've got lab technologists that are trying to control assays for compounds that you rarely see. So there's lots of issues that are associated with this. We have incidental findings. We have unexpected positives that need to be potentially followed up by the clinician. So there's lots of concerns in regards to just having bigger and bigger panels. And it also comes down to the idea of what is really proper utilization, right? Do we want to have comprehensive testing done on everybody? Because this is where ultimately it leads us to. And this is data from Dr. Catherine Smith, who is an R&D scientist at ARUP Laboratories. But this is what inevitably happens when we start thinking about using mass spectrometry, having bigger and bigger panels. Here's sort of an example of a comprehensive method, including over 150 different compounds, ranging from over-the-counters all the way through prescription medications like opiates, as well as benzodiazepines and others. And the question really becomes, is this what we want to do, right? Do we want to have these types of methods where everybody's being screened for everything all the time? There's a lot of expense associated with that. There's a lot of potential troubleshooting that needs to go into such a huge such a huge sort of screening method as well. And so it really presents a challenge. We're trying to have this one panel for everybody, but at the same time, that might not be the best approach we're looking for. So if we step back and we think a little bit about the technologies that we actually have, how we've been using them and how we might want to use them, it really sort of brings up, I think, the question that we're all trying to answer is, what are we looking for from an actual urine toxicology workflow? And so I do think that a qualitative screen to rule out can be extremely helpful, right? It allows us to do a very broad panel of drugs. We can still detect illicit drugs. If we don't think there's a very high probability, we don't really want to spend a whole lot of time doing high-level confirmatory type methods. And that's really sort of the root of doing a screen prior to doing confirmation, is we want to rule out a bunch of stuff, right? We want to do fast and cheap screens before we move on to more expensive mass spec-based testing if we can help it. So we still want to have that panel, we still want to have that large detection, but at the same time, we're trying to detect things like prescription compliance, we're trying to detect abuse. We want to have something that's very sensitive and specific, because again, just having a ruling out screen, if it's not very sensitive, well, then that's not very helpful to rule things out. Very classic example is 7-aminoclonazepam repeatedly screening negative because it doesn't cross-react well with the majority of benzodiazepine methods that are out there. So we need to have sensitivity and specificity that is appropriate. But at the same time, because we're going to be using these to rule out, we do want it to be cheap and we do want it to be fast because we want to decrease the noise and get to the signal that we're trying to actually look at closer. And we do want it to be comprehensive, or at least as comprehensive as is needed. And we want it to be scalable and flexible. We don't want to have to worry about small panels, large panels, setting up different types of methods. It would be very nice to be able to fully integrate all of these together. And then certainly one of the most important, I would say, depending upon your viewpoint, but for any of those who do direct laboratories, you want to make sure that it's easy, right? We don't want to have really complicated, difficult to control assays that are going to be problematic for the laboratory to maintain day to day. On the other side of that, we want a quantitative confirmation as well. And again, quantitative, you could argue back and forth between qualitative and quantitative. But I think it's very interesting to think about the percent positives of the results that do require confirmation, and whether or not we want to provide confirmation from the start, or whether or not it's something that physicians can follow up with, or the laboratory can in some way be triggered to help with confirmation as well. So from that, we want it to be sensitive and specific also. We want to be able to get metabolite ratios, and that's one of the powerful things about doing quantitative results, is you can get sort of true ratios. We can also, we also at the same time want to be able to distinguish pharmaceutical impurities, like seeing low levels of hydrocodone in the presence of high levels of oxycodone, and that being a pharmaceutical impurity, not somebody who's taking two different medications, one of which they most likely are not being prescribed at the same time. And then lastly, doing more complicated things like DL ratios for amphetamine or methamphetamine to try to distinguish the potential source of the compound. So now I want to move forward with the idea of, so we have all of this technology, we kind of have an idea of really what do we want from a workflow. We want something that's flexible, we want something that can have large panels, small panels, but we still do potentially want to think about having a screen that is available. So this is data that's from Natalie Rasmussen, who's another R&D scientist at AAP Laboratories, and she has been working on an LC-TOF rapid screen, which is what we're calling this. And this is really looking at the ways that we've been using time-of-flight technology and how we might be able to use them differently to be able to really get a lot more for what we're able to get from a typical mass spectrometer. So as of right now, what you can see here is a pretty complicated chromatogram. And as I explained earlier, this is one of the huge benefits of time-of-flight mass spec, is we can really pack a lot of data and a lot of peaks and compounds into a pretty small window, and we don't have to worry about missing anything or data quality. So here you're seeing a less than 1.2 minutes to actually cover every single immunoassay that we currently offer on our test menu in urine in less than 1.2 minutes on a time-of-flight mass spec. I think what's also quite exciting about this is we can get this to the point where the cost is nearly equivocal to a routine 9-compound immunoassay panel. So now all of a sudden this idea that we have to have these small panels, large panels is starting to fall away. And then lastly, I think that's even more exciting is we can do enzymatic hydrolysis, so we can boost sensitivity for compounds like the benzodiazepine class, where many of those are conjugated to glucuronides. And so we can get a really good sensitive, but at the same time, a very specific screen being done by time-of-flight mass spectrometry. Very quick, very cheap, nice way to actually start out. Looking at the analytical performance of this, I won't go into too much, but not surprisingly, we're using a high-performance instrument to do this screen. And so for that reason, we're able to confirm everything that the immunoassay is finding. We're able to reduce the false positives that the immunoassays are generating, but we're also able to find some things that the immunoassays were not able to find, particularly in that benzodiazepine class, where we do have cross-reactivity problems as well. And so what we're really kind of seeing here is the application of a better technology to be able to get better results, but not having to do complicated workflows up on top of that. I think the other thing that's really kind of exciting as well is we're able to do quantitative creatinine testing at the exact same time. So one of those compounds that I showed you in that complex chromatogram was in fact creatinine. And here you can actually see the performance of a creatinine assay. And so now we have a mixture. We have a mixture of quantitative testing for creatinine, qualitative positive, negative detection for all of the drugs included. The imprecision of the creatinine assay is sitting around 3% at 87 milligrams per deciliter, 2% at 230. You can see the accuracy overall is about an 8% bias throughout the AMR of 20 to 400. And it's kind of interesting to see how harmonized a lot of the creatinine assays are, but it doesn't necessarily mean that they are perfectly accurate. And so we are continuing to look at that 8% bias, even though certainly not unacceptable, something we are interested in looking at a bit closer. So what this really kind of brings in is the idea of then custom panels with confirmation. And so you can have an instrument like a time-of-flight mass spec that has the ability to look at 120 different things or more, but we don't necessarily have to look for all of those, right? We can pare it down to only look for 2 in a specific test if that's what's been ordered, but the assay itself is the same, right? So we don't have to worry about batching testing. We don't have to worry about setting testing up for panel 1 versus panel 2. and we don't have to worry about maintaining a larger panel if it's not ordered very often. Everything's run together, and so it's very exciting to be able to use a single technology to funnel everything through, but also have a very sensitive and specific technology that we're doing. And then certainly based upon what the panel was that was set up, even though everything's running through the same assay, we're able to direct it to confirmation in a much more intelligent way. We don't have to direct things to confirmation that the client wasn't looking for or the physician wasn't expecting to see. And so we can get away from having excess confirmation, excess reflex testing, and make sure that only what's being monitored and only what's of importance to the physician for decision making is what we're going to be following up with confirmation testing. So the last few points, making the complex simple, I think is what we're all kind of striving for. We want it to be simple from a physician standpoint, but we also want it to be simple from the laboratory standpoint as well. We certainly want to continue to design testing with the patient and the physician in mind, not reimbursement. And I think that's a challenge in this space, but it's something that we all need to continue to do together to make sure that we're not worried about how to use a technology because of reimbursement, but using a technology because it's really the right technology for the answer that we're trying to get. And that kind of goes to the third point of finding the right technology and putting it where it can actually have the greatest impact for the overall sort of patient care continuum. And then lastly, what I think, leave everybody with, and the reason I wanted to talk about this was to really just kind of stimulate some thinking around the idea of the future being in intelligently designed custom panels using advanced screening capabilities with tried and true confirmatory methods. And I think this is really going to be sort of an exciting area to see shape up and come together in regards to proper utilization, but at the same time being able to offer flexibility for physicians in the future. So with that, any questions, I'd be happy to take them now or offline as well. Here is my email if anybody would like to sort of send a question if you wanted to discuss these aspects, or if you had any comments, or if any of this sort of stimulated any kind of discussions that you might be having in your organization, I'd be happy to talk about that as well. Thanks again for your attention. I appreciate it.